Okay, good morning everybody. Let's get started. So our speaker today is Fernando Peruani. He's coming from a place you probably all know from your vacation. He's from the French Côte d'Azur, and you cannot only do nice vacation there, you can also do very nice science there. Let me just briefly introduce him to you. Fernando grew up in Argentina, where he studied physics at the University of Buenos Aires. Then he went to Germany, to Dresden at the MPI for complex systems, where he was doing his PhD on simulations on self-propelled active particles which mimic the behavior of living systems. Then he went as a postdoc to the University of Saclay. He also became an invited professor at the University of Buenos Aires, and then was appointed as a group leader to the Center of Integrative Biology at the University of Toulouse. And actually, I don't know since when you are in Nice? Since
2011.
Okay, so since 7 years he's now in Nice, where he is an associate professor and having a group on complex biological systems and active matter. So by training, Fernando is a theoretician, but he also is doing experiments in his group on sheep, on bacteria, so he covers the entire length and time scales. I should also mention that he is, in addition to what he will tell us about today, he's also working on the spreading of information in moving agent systems. He's also looking at the proliferation of diseases, for example, HIV infections. But today he's talking about the intermittent behavior across scales in biology.
Okay, so thank you for the introduction, Clemens, and for the invitation. Thanks for you for coming. So, in fact, I was planning to introduce a bit what I do, so I will start this way. So what we do there in my group, we work on collective behavior in living systems. Mainly the focus is on modeling, but we also do some experiments. Currently we are working with bacteria, with ants, and with sheep. And what we do in our research, basically the way of telling it or summarizing it is we propose equation of motion or behavioral rules for the individuals, being those bacteria, ants, or sheep. And we try to check whether this equation of motion or behavioral rules account for the observed collective patterns in these systems, with the intention, probably many of you do that here, is extracting the self-organizing principles. And we do that, basically this is to tell you a bit our approach. We try to look at that in experiments, using experiments to characterize the behavior of individuals, to characterize as well the behavior of a collective, of a group of individuals, but we also play this game performing simulations. Basically, that means that we propose equation of motions for the individuals, we simulate it, and we try to see what happens with the collection, a large collection of these individuals. But we also play this other game that is a more theoretical game. Basically, I'm sitting in math, so that we play, and we have to be a bit more theoretical at that level. So we apply constraint techniques, and basically with the intention of describing the macroscopic behavior of these systems in terms of few microscopic variables. And why we do that? This is an example, for example, of an active system of particles with nematic alignment, and it's because you may try to understand the collective behavior that you observe in terms of instabilities and bifurcations. That's why we play as well this game, right? We take also this more analytical branch. Okay, now, I will try to avoid as much as possible mathematics or equations. Some may appear anyway. I'm going to talk about intermittent behavior, mainly intermittent motion of Escherichia coli and sheep. And I will try to convince you that we can look at these two systems using similar mathematical tools. Systems are very different though. Okay, so bacteria. So with bacteria, the first thing I will try to convince you is that if you think of pathogenic bacteria and if you are thinking of Escherichia coli or Salmonella, they are they are flagellated bacteria that swim, and even though they have been studied quite a lot in 3D, meaning far away from walls, they live close to surfaces, or there is an interest for them to be close to surfaces. So let me start with a pathogenic bacterium like Escherichia coli, one of the pathogenic strains. You swallow food that is contaminated, that goes through the digestive system, but at some point the bacterium goes through that tube, has to come close in some areas of the gastrointestinal tract, have to come close to the epithelium in order to infect some cells. So at some point, what we can say is that basically near surfaces, the level of nutrient is higher than in the bulk, plus that on surfaces, you find the cells that this bacteria may infect. That you may do it in vitro experiment, but otherwise you can think like a tissue, like a surface in itself. So that's two reasons why for bacteria it's good to be close to surfaces, and that's why that represents an advantage to them. That will be central for the rest of the talk. Now let me summarize a bit what happens with Escherichia coli far away from a surface. So if bacteria is swimming far away from a surface, so the motion of bacteria is, typically it's called this type of motion run and tumble. And in this cartoon, this is sketched in this way. So bacteria move in a straight trajectory and then perform an abrupt change of direction called tumble. And again, it's going to move straight, another tumble, and so on. However, when they come close to a surface, we see that the motility pattern changes, and changes in this way. For example, here you start to see that the trajectory is not more, it becomes smoother, and in fact turns to be rather circular. And there are two reasons for that. When they are close to a surface, hydrodynamics produce an effective attraction, if that the bacterium towards that surface, and there is another important effect connected to hydrodynamics. The bacterium, you have the bacterium that has the flagellar bundle that is rotating, and so the bundle turns in one direction, the body turns in the opposite direction, and that produces a torque, effectively a torque, which forces bacteria, if we are looking at the bacterium, if it's close to the surface, to move in circles. So basically, as they come close to a surface, they are going to get for a very long time close to the surface and will start moving in circles. Okay, now the bacterium I'm going to talk about is one special strain, a very pathogenic strain of Escherichia coli, which is called enteromorrhagic, and that's the name because it causes bloody diarrhea. And this is the bacterium that was produced an outbreak in Germany, something like, I don't remember the year, 2-3 years ago, a bit more, and 300 people died. It was, I think, contaminated asparagus. And another reason to study pathogenic bacteria is that by 2050, basically infections due to the antibiotic resistance, the number of people dying due to bacterial infections will surpass those related to cancer. So in 20 years or so, bacterial infection will become more important even than cancer today. Okay, but let me show you how it's happening with Escherichia coli. As I said to you, as they come close to a surface, and that would be glass, what you can see is that bacteria start to move in circles in this way. So this is an experimental, this is the experiment. So the bacteria is moving around, and as you can see, it's basically performing not perfect, but a circular trajectory. So you may ask if this circular trajectory is related to hydrodynamics. Basically, hydrodynamics is forcing bacteria to revisit the same area in space, so it's turning around in this direction. So let's suppose that there is an interest for this bacterium to locate eventually something that is maybe located on the surface. That could be a cell, a host cell that we do experiment where we leave host cells on the surface. That could be eventually that there is a source of nutrients. But anyway, if we think that there is an interest for this bacterium not to get trapped and explore the surface, how they can do that? And this is what I'm going to show you now. So this was an example where the bacterium didn't perform any stop, but often what we observe are these stop events. That is what you saw here. So basically, I summarize in this way. By performing this peculiar stop event, bacteria manage to escape from these circular traps and set a new moving direction. As you can see here, it's trying to move like in a circle, makes stop and resume moving in some other direction, but again, due to hydrodynamics, will be turning in circles, perform another stop, and by doing that, manage to move way more than in this example. So, and this is what we are going basically to see in a bit more detail. So because bacteria is performing these stops, what you can do is you can look at what happens with the speed of the bacterium as function of time. That is what you see here. This is the speed of the bacterium as function of time. So this quantity, the speed, is showing an intermittent behavior. So it's high, was around here, drop the speed, up again and down and so on. So if you look at the speed distribution, which is this figure in here, you can see that it's a bimodal. So you can try to define the local minimum here to define a threshold. And now we are going to say that if the speed is below the threshold, we are around this area, we are going to say that the bacterium is in the stop regime. And if the speed is above this threshold, we are going to say that the bacterium is in the running regime. So why to do that? Because we are interested in looking at what happens with the distribution of this time, which I'm going to call the runtime and the stop time. So we want to know the distribution of these two, and I will show you in a few seconds why is that. So what we want to know is the distribution of this, and here what you see is the survival curve associated to this distribution time, meaning this is the accumulated distribution. If you look at what happens with the runtime distribution, and this is log ln, what you see is a straight line. And if you look at the stop times, what you see is something different. Something different, which is clearly not straight line in here, so it's not an exponential. So let's assume that we have two states, the stop regime and the moving regime as we defined before, and we assume that these basically are two behavioral states. So either the bacteria is in the moving phase or is in the stop phase, and then we have transition rates from one state to the other. If that would be our hypothesis, then the runtime distribution is consistent with this picture. We will get an exponential distribution for the survival curve of the runtime distribution, but this is not consistent with the distribution that we observe here. That should be, again, a straight line, which is not. So let's suppose that we insist with this idea that we can describe the behavior that we observe with the Markov chain. So meaning that we have to define a number of behavioral states and arrows that connect those states. So if you assume that still you insist that this data may be represented by a Markov chain, that can work, but then what we need to do is to include one extra state. So we will need 3 states, 2 of them associated with the stop regime and 1 with the moving regime. So, and in consequence, we will have 6 arrows. If you assume that, I'm going to show you next, you can explain these observations. Now, the question is, if we have these 2 states that you can deduce because of this way of reasoning, you look at the distribution of these times and you try to ask yourself what is the minimum Markov chain that explains the data that you observe, we need to include this. I mean, we, instead of having just one state, one behavioral state associated with the stop regime, we have 2. Now the question is, well, can we identify these 2 states in the experiment? So we went back to the experiment and we started to look very carefully what was happening each time that the speed was below the threshold. And this is what we observed. We observed that very often the bacteria was swimming, that you have one is with the tracking and the other side is without tracking. You will see that the bacteria is going to do a stop, but immediately after the stop, something is going to happen, which is this. So what happened there is the bacteria was moving at some point, made a stop, anchored to the surface, and start to rotate around that point, and at some point manage to release the addition to the surface and continue moving. So these kind of situations you can identify if you look at the curvature of this curve. I mean, probably you saw that it's relatively easy to identify that behavior. And now we have a way to identify the three behavioral states. One is going to be running, basically what we saw at the beginning as the bacterium was moving at relatively high speed. One means that it's in this tethered rotation, this kind of motion that we saw here, and 0 means that simply is not moving. Okay, so now since we can identify easily these three states, what we can do is we can look at one of these movies and basically check every time, I mean at every instant in which state the bacterium is. And then you can define when it's state 2, when it's state 0, and when it's state 1, and when it's transition from 1 to 0, 0 to 1, and so on. That this is, okay, probably you wanted to see that, I don't know. I mean, basically you will see a lot of time that the bacterium is performing this kind of tethered-like motion, and then we'll resume moving. Okay, so as I said, we have this idea, I mean we identify three behavioral states, and now we want to obtain the rates that associate all these arrows. So what you can do is you can measure the time the bacterium was in state 2, 1, and 0, the distribution of these times. This is represented again as a survival curve, and these are three exponentials that you obtain here, and due to the kind of, I mean, by analyzing the data, you can also check how many times the bacterium was moving from state 0 to 1, from state 0 to 2, from 1 to 0, and from 1 to 2, and so on. Basically, these are these Q elements. If you have this kind of data and this, you can define all transition rates in the system. So we can go to the data, we identify all transition rates, and now what you can do is simply put all this together and in a circuit, because of two of them are basically never occur, or one very rarely. So basically, instead of having 6, we end up having 4 transition rates that are relevant here. And what you can do is you take all the data that we measure from the experiments, and now what you can do is this straight line that in fact was before, and the blue line are the result of making this measurement, as you can see, work very well. So basically, once you have this, you have a Markov chain, and then you can write the associated master equation, and from the associated master equation, you can obtain this kind of distributions. Okay, so we want to do something else. I'm not going to enter much into the details. I'm happy to talk about the details if someone is interested. But what we are going to do is we have a Markov chain that basically tells us when the bacterium was going to state 0, 1, and 2. And now what we can do is use this model, this Markov chain, and plug it in an equation of motion where there is the speed and this omega that basically depends on the behavioral state. The evolution, temporal evolution of I is going to be given by the Markov chain, but the position of the bacterium in space is going to be given by a Langevin equation, in fact by 3 Langevin equations if you want, because this is changing as I is changing. So V can be 0 or it can be VR, and the same with omega. Now, the good thing of this is that now what you can do is you try to describe what happens in the system using a probability of finding a bacterium in position X with moving in orientation theta at time t and in state I. I mean, this is not very important, but what is important is that you can do the calculations, and basically you can use the Markov chain to generate a model that describes where the bacterium is in space. So you do that, then what you can do is you can write an equation for this. It's an ugly thing because an integral partial differential equation. This is a forward Komorov equation with transition chunks. But that you can solve, I mean, work with these equations. And then what you can do is use it to define the diffusion coefficient of this bacteria. And the diffusion coefficient is going to be a function of the parameters of the Markov chain. This is basically what I wanted to say. We went to a Markov chain, we do some calculation, and we end up having the diffusion coefficient as function of the parameters of the Markov chain. The diffusion coefficient is going to tell us, tell us how a bacterium explores the space. If it's large, means that it is able to explore a large surface of the space. If it's very small, means that it's basically exploring a very little area in the space. So now what I'm showing you is this function, the diffusion coefficient, as function of one of the parameters of the Markov chain, which is this transition K20, that is moving from state 2, what was motion, to state 0, that was just stop. So this is the key parameter in the model. And as you can see, this is the equation I showed you before. There are the simulations. And basically, there are two limits that are easy to understand. Basically, if this K20 is very large, that means whenever you visit here, you immediately go to state 0. That means that you stay very little time moving. And if you stay very little time moving, what happens is your diffusion coefficient is very slow. I mean, basically, you are not moving or moving very little. On the other hand, if this K20 comes close to 0, means that whenever you came in 2, you never come back to 0, meaning you will end up trapped in state 2. You don't perform any stop. If you don't perform any stop, what happens is you get this circular kind of motion, and because it's not a perfect circle, what ends up happening is that you may explore the space, but you will explore the space in a very inefficient way. Basically, you stay around the same position in the space, same area. Slowly, you will start exploring, but very slowly. Now, the question is where the diffusion coefficient of bacteria is located, because as you can see, there is some intermediate value. If this rate is too large, it's bad. If this rate is too low, it's bad again. But as you can see here, there is an optimal value that maximizes the diffusion coefficient, which means that if bacterium stop at that frequency, will basically perform the maximum exploration of space possible according to the model. And the experimental values are there. That's why what is interesting here. So these guys basically are performing these stops. They have the frequency at the optimal frequency according to the model I show you. And now you may think that this is kind of a coincidence, but then you can go and look at different type of bacteria, bacterial strain, bacterial species if you want, and on different substrates. And basically we saw that there is one example, but there are several others. And what we observe is basically the three-state idea works in other bacteria. And then there is also an optimal value at the value estimated by the model. So this seems to be rather general and not specific for the EEC, which is the enteromorrhagic Escherichia coli. But there is an important observation, because it's true that the bacterium may maximize, so this is the curve again, the diffusion coefficient as function of K20. This is the frequency at which you perform the stops. And as you can see, that depends on this KST, which is a combination of the other parameters in that Markov chain. All those parameters depend on the properties of the surface and on the properties of the adhesion of the bacterium. So if you have a surface where the bacterium will glue and will not be able to detach, that won't be a good idea to attach to a surface. The bacterium will get glue in an irreversible way or for a very long time. On the other hand, if the bacterium is very weak, the adhesion will not be able to perform this turning I showed you before. So basically will get there, will stop, but will continue moving in the same direction. So will not be able to escape from these circular trajectories. So depending on these properties, you may be in one of these curves, and then it's a very good idea to perform stops. So by performing stops, you will increase your diffusion coefficient. But if you are in this extreme situation where the adhesion doesn't work or works too well in the sense that cannot detach from the surface, it's no longer a good idea. And then we are in curves as the purple one or the green one. And in here, the best strategy is not to perform any stop at all. Right, you understood? So it's often that means the best strategy is to perform stops, but this is not wherever bacterium you look, swimming on close to a surface, and whatever surface, you will always observe what I have shown you. In a large variety of systems, yes, but in some others, the answer will be not. Okay, so with this, I concluded, I mean, this is the kind of what we have learned, I would say, from this, is that bacteria explore surfaces by performing transient addition events. So something important is on top of the hydrodynamics that is typically studied, one has to look what happens with addition to surfaces, and that plays a major role. And I show you that there exists an optimal stop frequency that maximizes the diffusion coefficient. And now the rest is way more speculative. I mean, the speculation is that the optimal frequency of stop is close to the basal frequency observed in 3D for bacteria. And in 3D, there is nothing special with that, the frequency in 3D for tumblings. So this is not optimizing anything, but as you squish that kind of motion and you go to 2D, to a surface, you see that now you can get kind of optimal transport, and you can do that by regulating the stop frequency of the bacterium. And I should say that a bacterium is able as well to sense in which surface is moving, and there's, I mean, indication that there is a feedback from the surface to the flagellar motor and so on. So it is apparently, potentially, bacteria is able to regulate at will, let's say, the stop frequency. But with all that, basically, the message is that probably bacteria have evolved in order to become excellent surface explorers rather than 3D swimmers. Each time that we look at papers for bacteria and thinking in evolution, most of those papers will try basically to think of the shape and the properties that we observe in order to optimize, for example, minimize the dragging as they move in 3D and so on. But I think most probably these guys have evolved in order to perform very good exploration of surfaces. Okay. So, well, where do we go with this? Now what we are doing is this. We are trying to understand how bacteria search for host cells and infect them. So in the case of Salmonella, what they do is they enter inside these cells. These are epithelial cells from the colon. And the bacteria hope that you manage to see. They're moving around, and eventually they manage to get inside the cell. If it's Salmonella, if it's Escherichia coli, they will get anchored there. Here you can see, probably not much, but that the bacteria are hitting and basically trying to infect the cell. And then we are coming up with models that basically try to mimic this, and we are trying to exploit and put forward the idea that the bacterial infection involves self-organization and collective behaviors, and basically extending the model I showed you before. Okay, so sheep. Now I change. We forget about bacteria. We move to sheep. And here what you see is an arena of 80 meters by 80 meters. Pictures are taken from a tower that is, the shadow is here. And I hope that you managed to see that what is happening here, basically these guys are spreading and they are contracting. And then you will see this again. So they spread and contract. And then if you look at experiments typically, I mean, we were looking at sheep over 2 to 3 hours, and some a bit longer, 4 hours, but not more. And typically this is the behavior that you observe over time. So spreading and contraction. So how can you characterize this type of motion? So that what you can do is, let me go. You can look at the surface covered by the group, a function of time. This is the convex hull, and basically the area of that convex hull. And then what you can see is that area covered by the group increases. That was when we were observing the spreading phase of sheep. Now this area was increasing. And suddenly we saw that there was a contraction event. This is when this the surface covered by the group drops, and so on. So as you see, again, we have an intermittent motion in the contraction. This time is collective, of contraction of the surface covered by the group. Now, what you can do is, if you want to model that kind of stuff, which we have done, I mean, in this paper, what you can do is you can use a kind of ad hoc Vic-Check model. That is what we use. I will be forced to criticize a bit what we have done, but anyway. So this is what we have done, a Vic-Check-like model. If there is time, I will say a few words about that, but if not, I mean, the only thing I want you to know is that it is possible to get this kind of behavior, but in order to do that, what we needed to use is a model where parameters cannot be obtained from the experiments, which typically is bad. And due to the problems of tracking, as you saw, the tracking was taken by, I mean, we got the tracking of the position of ship by taking picture from a tower. The thing is that we got the position of ship every minute, but not every second. We couldn't, the tracking of every individual was not done. We couldn't do that. And then basically, due to that, most of the parameters that we use for this model were basically tuned in order to get something that mimics the behavior that we observe in the experiment. The model is reasonable, and by tuning those parameters, you get something that looks similar. But, it's not satisfactory, I would say. So we decided to revisit the problem. I mean, and we look at experiment with smaller number of sheep, 4. And 4, 3, and 2. And this is what you observe. So these are these 4 guys here. And as you can see, they are eating. They stop. They move. They move. Whenever they move, you hope that you see that they form basically a file. I mean, this is one sheep after the other. They are again not moving much there. And if we wait a bit, they will start moving. Well, some hesitation. Now they start moving again. I mean, we want to characterize what we have observed. And the idea was, it's going to be, I mean, the intention is to connect it to what we saw with bacteria, with Escherichia coli. And we will do something similar. Because here what I'm showing you is the speed of ship 1, 2, 3, and 4 of the previous movie. I mean, this is speed as function of time of the 4 sheep. And as you can see, with the speed happens something similar. Ship 1 is moving, suddenly stop moving, moves again, stop moving, and so on. When there is something that becomes evident, whenever you see that ship 1 is moving, ship 2 is moving, ship 3 is moving, and ship 4 is moving. So basically there is a high degree of synchronization between them. And now a bit more technical comment. What you can do is you can look at the speed distribution that you get from this time series, and you can define this point in here around 0.5 to define, as we did with Escherichia coli, a threshold, and say if this is above that value, we are going to say that the ship is supposed in the moving regime, and then we put a 1 here. And if it's below, we say that it's in the non-moving regime or stop regime, and we put a 0. And as you can see, when this guy is moving, here we get a 1, then was silence we get here, then there is a peak, we get a peak, there is a second peak, we get a second peak, and so on. So the method works relatively well. We can go from this to a binary signal. Now, from this binary signal, what you can do is something similar to what we did before. You can take the signal and you can look at the time this quantity adopts the value 1, so which means the duration of this moving regime and the time that was non-moving. So basically this is going to be the same strategy as before, and what we are going to do is we are going to look at the survival curve associated to the moving time and the non-moving time in here. And this is what we get. This time, what we get is that for runtime distribution, this was plotted in lean because this guy in here. But if you look at if you look at the distribution of runtime, what you get is again an exponential. And now, if you look at the distribution of stop times, what you see is something that is rather a gamma-shaped distribution. Again, this is if we would be thinking in having 2 states as before, one behavioral state that would be moving and one behavioral state that would be non-moving, we will get that this curve in here should be like the blue one, should be as well as an exponential. So again, here we will need to introduce 3 states in order to explain what we are observing. And these 3 states are going to be moving, and 2 are going to be associated to the stop regime. That is what you see here, right? I mean, in order to get a gamma shape, you can get it at least having 2 states or more, but with 2 is going to work. And now the question is, if we postulate the existence of these 2 behavioral states, what are these states? Again, right? I mean, this is coming from analyzing, a time series, and then you go back to the data, and what you can do is define this state in the following way. You could say, when we get one state that is easy, the guy is just moving at a relatively high speed, but then there are 2 situations that are easily to spot, to identify. We have the chip is not moving, and when they are not moving, typically they are either, we see them with the head up or with the head down. With the head down, this is an interpretation, this is what we will use, basically head down or head up of the ship. Head down, we will tend to associate with grazing, and in fact, when you look at the experiments and you see these guys close to you, it's obvious that they are eating, typically when they have the head down. We don't know for sure if every time they have the head down they are eating, but we can be sure that when they have the head up, they are not eating, at least they are not getting grass from the ground. Okay, so now that you have this, you can again define all transition rates. As before, we have 6, well, represented slightly different way, but it's 6 again. And basically, you see that there are 3 of them that are basically dominant. We consider all of them, but here we will show you a simplified version of the whole story, which I will keep only 3 of them. The ones that are more prominent. But we took into account all of them as well, in the analysis. And okay, so, but let me go with this simplified picture. So, this is what we will finally be using from one individual, but as I show you, the difference with Escherichia coli is that now these guys, they show a very, synchronized behavior. And if there is a synchronized behavior, something should be happening. If each of them have this kind of small Markov chain in their brain, let's say, so they have to be talking to each other in some way. And the key idea to get to the highly synchronized behaviors that we observe is to assume that the rates of each individual depend on the state of neighboring individuals, right? So each individual will know in which state is the other, which is not that difficult to realize, because if someone is moving, it's easy to realize that it's moving. If someone is with the head down, you will see the other with the head down or the head up. Okay, so now in order to get to how this behavioral, how these rates are, what you can do is you can look, you can perform experiments with trained sheep. Sheep you can train. They may not be the most clever animals on earth, but they are not that stupid, and they can get this kind of association. Each time that the collar vibrates, the guy will know that the yellow panel will appear somewhere in the field, and whenever that yellow panel appears, will come close because it's going to get a reward. Food. So you perform that a lot of times, and one day the guy will start to really, will learn and will start moving to the yellow panel, and then you have a trained sheep. So then what you can do is the following. You get a group of naive sheep, they are there, and there is this trained sheep, and what you do is you send the signal to the collar, vibrates, you show the yellow panel, the guy starts running towards the yellow panel, and the other guys start following. And then what we want to do, in fact, the goal of the experiment is to characterize with what probability, as function of time, this guy will stay with the other guys that are not following, and with which, if decides to stay or start to follow the others, right? So basically, that's the idea. Should I stay or should I go? And okay, so with this, what we got is basically the transition rates have this functional form, or that this functional form, explains well this experiment I show you. And basically, there are 2 contributions. Basically, you are in state I and you want to know with what probability you go to state J here. This one, I mean, that means that you are in state I. If you are in state I, you are inhibited by the guys that are in your state. I'm not moving, and basically many people around me are not moving. I will be rather inhibited from moving. However, if there are many moving, this is, the excitatory part or the recruiting kind of term. The more I'm moving, the more prone I'm going to be to start moving. And there is something else that I'm not addressing here. If you have this kind of transition rate and so on, what you can see is that in fact there is, you define a collective decision-making kind of mechanism. Because if you have the guy that is the leader start moving and goes to the panel without managing to convince anybody, this guy will not manage to move the rest of the group. So, there is a period during which this guy will be able to promote a transition to the moving state. Anyway, so now if we go back to the experiment I showed you with the force-ship, you can use the same strategy and you can use basically the same model or the same functional form for the transition rate I showed you before. How can you work with with the force-ship? You do something similar. What you can do, you look the spontaneous behavior of this force-ship and then you count for how long the force-ship were all of them not moving, when 3 of them were not moving and 1 was moving, and so on. By looking at the statistics of this plateau here, with this curve, basically you can fit all the parameters. And this is a kind of technical issue, but you can do by looking at this kind of curves. And if you do that, the main difference now, because there is in the force-ship there was no leader, or not ship with a vibrating collar, then there is a spontaneous transition rate of transition from one state to another. And this transition rate, as you can see here, in fact, is not just a constant. I could have glued it with this. So in fact, there is the spontaneous transition rate from one state to another is modulated by the size of the group. That is, in fact, what is happening. So there is an influence of transition if nobody is in that state of how many are in my state. Anyway, so this is what you one needs to put into the model. This is, again, I'm going to skip it, but basically, once you have that, you can define a master equation, and you can do a kind of Cramer-Moyal expansion, and why you may do that. This is only valid for large n, but the interest of doing that is if you have all that, what you can end up showing is that the system behaves like a self-excitable system. That was the thing. Basically, you can define this in effectively 1D system where there were 3 unstable fixed points and 3 stable points and transition from the stable points. And basically that this defines a collective decision-making mechanism. And that you can show by looking at this kind of analytical treatment of the system I show you. But now what we can do is something similar to what I showed you before. What you can do is you can look at basically what we got is a model for how the states in the behavioral state of chips are basically transition, but now we have to do, if we want to describe how they move in space, we have to couple this to an equation that describes where the chips are in space. So we need an equation of motion, and the equation of motion is going to be a function of the behavioral state of the individual. That you can do, and if you do that, basically what you obtain here is just the speed that is changing, and okay, this is working. Basically, you get here this guy's moving. This is the position in space. This is, the mean square displacement, and this is how many ships are moving at a given time. So you saw a collective movement in there, and the same is happening in here. And basically, this is the experiment, and this is the model. And what you get is something that is quite comparable. For most quantities that we can measure, the model seems to be consistent with the observations. Okay, so we get a model that more or less describes how these guys move, but where we can go with that. And now comes what we did as well. So these guys are basically eating grass, so they are not only exploring the surface, they are exploiting it. So they are exploiting the area, the environment where we put them. So you may wonder whether that is for example, where this is an optimal way of eating grass or exploiting the resources they have around. So what you can do is you can put an equation for the grass that you have in the system, and you can put an equation basically that takes into account that every individual is ingesting a given value, number of grass in there. So, and then what you would get is basically a guise of that will maybe moving around in a system that is basically, you may replenish, basically you may assume that they never manage to eat all the grass around, or you may assume that they may be depleting areas as they move around, as you can see in the middle, or this other situation that is a more extreme scenario where basically they will never grow again grass. Now, if you play with that, what you can see is that there is a range of parameters, which coincides with the values that we measure, where there is an optimal strategy. We can prove that there is an optimal strategy, and we can prove that there is a window of parameters where what we observe would be this sheep would be basically sitting, behaving, performing this optimal strategy. That is what I would say. It's not for all values of grass that you will get that this is optimal, but it's a range where this would be true. Okay, so from this part, what we have learned is that intermittent collective motion requires 3 behavioral states, that parameters are such that the system becomes self-excitable, and that there exists a link between intermittent collective motion and collective foraging. That was the last part I showed you. And I show you that this collective foraging, this is what we can show, that there exists an optimal collective foraging strategy, and that there is a window of parameter space that coincides with what we observe in the experiments. Okay, so with that, I will summarize all this part. What I have shown you is intermittent behavior in 2 systems, bacteria and sheep. In both cases, in order to analyze the data, what we did is we converted an experimental signal in a binary signal. Then we studied the distribution of time the system stays in state 0 or 1. And then from that, what we did is having those distributions, we basically use it to get how many number of behavioral states are present in the system, and then we we analyze the resulting systems of equation. And we found that for bacteria and sheep, to display intermittent behavior, we need 3 behavioral states. Now, we stick with sheep, but let me start by something that if you have played with the big sheep model or flocking, you may know. So I'm going to define or to recall you, that there are 2 alignment mechanisms that you can use. Now we are thinking in the moment where sheep were moving together, or birds, or fish, or whatever, and now there is 2 simplified versions of looking how they may move together. One version would be you focus on this red guy, and this we will implement a rule that we will call velocity-velocity alignment, which is what has been used by Reynolds, Vicek, and Cousin, and many others. So you have this situation, and this guy basically what will do is look at the velocity direction of these neighbors and basically align its moving direction with respect to the neighbors. If you do this kind of thing, basically it doesn't matter where you put the guys. These neighbors may be here, down, to the side, or to the other side, and basically the resulting moving direction is the same. All right. So now you can do something different. You could say, okay, we will ignore in which direction these guys are moving. Basically, we will just take into account where these guys are located, and we may implement a rule where this guy, instead of looking at the velocity, looks at the position. That's why we will call this position interaction or alignment. and this guy will basically move in the direction where most particles are in the interaction neighborhood. All right. So if you do that, the only thing that matters is where these guys are located, but not in which direction these guys are moving. Now let's go one step further, and now let's assume that the interactions are not reciprocal, which means that, suppose if I'm moving here and I have someone behind me, I have no idea what this guy will be doing, but this guy will look at me. So this guy may be imitating my behavior or not, but has the potential of being interacting with me. But if it's visual interaction and it's behind me, I cannot see the guy. So that would be a non-reciprocal interaction. So let's look at what happens if you have the velocity alignment, the classical one, and you put this kind of non-reciprocal interactions in pairs. This is what you would see. The guys will basically end up moving in a flock in the same direction. But if you do the same with the other one, position alignment, this is what you would see. So they also will get a polar state. Everybody is more or less oriented in the same direction, but they form a file. I mean, this is not the same. You see that there is clearly a structure in space that is very different from here. And now you can look at what happened in sheep. You already saw that in the experiment, or I mentioned to you. So these are the experiments. And here you have 4 guys, and basically they move one after the other. So basically, they are not doing velocity alignment, but position alignment. Or the data is not consistent with velocity alignment, I would say. You really need to get rid of that and use just position alignment. So from this part, basically, the message is that velocity-velocity correlations that has been systematically measured in experiments and in theory, but here it has been, in order to create those velocity-velocity correlation, typically it's assumed that there is a velocity-velocity alignment, which makes sense. But you can also do the same if you use a position alignment mechanism, so meaning that you don't need to invoke a velocity-velocity alignment, which means, well, this is a bit provocative, that one may go and revise every paper that has been used velocity-velocity alignment, because there are other options of getting a polarized flock of guys moving in the same direction, not invoking velocity-velocity alignment. Okay, so and with that, I will thank the guys that helped me to do this. I start with bacteria. This was a master student with me. Stefan Otte, this also Emiliano Perisipini, a postdoc with me. Then Luis Gomez Nava was working with sheep, in the modeling of sheep. Sylvain Toulet was doing most of the experiment I have shown you. And Lucas Barberis is connected to the last part. And in fact, with the person, I developed this model with the cone interactions and so on. And these are the biologists that have been working with me in the project I show you. Dorota Sheruka from Monaco and Richard Bon from Toulouse. Thank you. You can combine both, but there is something that comes from dynamical systems that I think is the most satisfactory answer for that, which perhaps is not the one that you want. But let's suppose that you add both. You can add both. Now, if you look at the equations, well, probably I better don't write it, but you have one equation that will contain the sine. I do it, I don't. I better don't, no? I mean, so you have an equation that is the difference of the two angles, plus another one that is going to be the relative position of the other and your angle. The fixed point of that equation, if you have just the second part, is also going to be that the two moving directions are the same. So it is redundant to add another equation because the system of equation will have still the same fixed point. So you can still fit the data using two. You may improve slightly the data that you may have, but including that alignment. But at the end of the day, you are increasing the number of parameters in your model and just improving very, very little the accuracy in the description. So you understand. Yeah. Right. So the answer is long, but there are a few things. First, in sheep, as they start, if you assume that they are depleting the grass, that they quickly become as well a patchy kind of environment. You could go that way, right? Because if I start eating and in a time scale that the grass doesn't grow, that is also going to get rough the landscape, and so you will get patches of grass here and there, but not everywhere. But let's focus on bacteria. Bacteria, what you have is these cells that are, epithelial cells that bacteria may want to infect. That seems to be the biological goal of these guys in order to reproduce. The problem, first thing, do they have chemotaxis? They are attracted towards these cells. We assume that was the case, but the statistics shows that it's not happening for whatever reason. Dorota is a bit, not happy with that. All other biologists were also saying that there is chemotaxis, but nobody really checked and did the math and the statistic to check whether chemotaxis was really helping them going towards the cells. We did it. We blindly tried to check and measure that, assuming that was there. But the statistic is absolutely not consistent with chemotaxis. So these guys are looking for the cells kind of blindly. We don't see any biased motion towards the cell. So that exists as well. You can do a random search without using signaling. However, there is signaling that occurs with the cells because whenever one bacterium, if it's Salmonella, gets inside the cell starts to secrete some chemical signals that will start to be produced that will basically warn the immune system as well as other bacteria. So once that takes at a longer time scale, but at some point the cell has to become and start to secrete the chemical that the bacteria may use to guide themselves and come to the cells. You understood there is a first stage. At the beginning of the infection, there is no chemotaxis. But at the end of the infection, everything indicates that chemotaxis is acting. So, but this is when the cell is really like with a lot of bacteria around and close to, it's dying basically. So that is where there is a second way of infection where the bacteria will know in which direction to go, or the dynamics is very different. That answer?
Yeah.
Let me say the different way. If you have a surface, what happens is hydrodynamics becomes a kind of handicap for a bacterium. If you think nature may have evolved in such a way that have a way of avoiding getting trapped just because there is any surface. There is a surface, hydrodynamic will tell you that you stay for a very long time. Now this has been measured, for example, in the group of Eric Clement. They can be they measure up to, I don't know, 40 minutes or something like that. That's the model. Kind of extreme, but 40 minutes moving, basically rotating in a spot. So that doesn't seem very good. So they have to find a strategy to not do that. And what we basically observe is addition to the surface may be the way of escaping from these circular trajectories that represent basically handicap to the bacteria or a problem due to hydrodynamics. Now there was another thing that I comment that these guys should be able to basically test the properties of the surface. That's not something that we have done, but other groups have done. Basically, they believe, and there is, I think, clear evidence that because of the adhesins and so on, that trigger cascades inside the bacterium. The bacterium basically is analyzing on which kind of surface is moving or is anchoring. And once, if it's a favorable situation for the bacteria, we may assume, I mean, when they are infecting E. coli at some point get anchored and doesn't move anymore. So because what it's going to do is just start secreting stuff to get control of the cell. And Salmonella, that's something similar, gets control of the cell by getting inside the cell. So basically will convince the cell to make a vesicle and get inside the cell. Right, I mean, this made them stay in 2D, but also move in circles. So addition is to do this kind of motion. I move around having an anchor somewhere that allows me to move, I mean, otherwise I may be trapped in this. So I start moving this way, not moving the large circular motion, and I will release, and I can move in a different circle. I'm going to be still in a circle, but then I stop again, and I do the same trick over and over and over, and then I manage to move, to explore the surface. Okay, so I think that there is an answer. I think that you can encode memory in a Markov chain. That is, I mean, we did what, because we wanted to identify the behavioral states. But if I wouldn't have gone to the experiments, which as a modeler I would be almost tempted to do, because then I say these two states that were S1 and S2 in bacteria, I just is a hidden state for you. That becomes immediately memory because it's not an accessible state for you. You understand? Yeah. But if you go internally, most probably it is even for us when we are using, I mean, if we have access to memory, we are in a state where we are having access to memory. So memory always, memory you can consider is always a situation where you have a larger system in complexity where you are lacking information, then you are invoking memory. But otherwise, you can keep on growing the dimension of your system and being back to a Markovian kind of situation, a non-memory one.